For decades, the story was simple. Modern humans wiped out the Neanderthals by force or outsmarted them. But systematic trauma surveys find no archaeological evidence for massacres, and combat injuries appear in less than 5% of Neanderthal remains, with no mass slaughter sites ever found. Instead, DNA analysis reveals that Neanderthals maintained a tiny effective population size from 1,000 to 3,000 individuals, making them acutely vulnerable to genetic drift and demographic shocks. Even more striking, up to 3.7% of Neanderthal genomes is modern human DNA, proving long-term contact and reciprocal interbreeding. This overturns the idea of sudden replacement and forces us to ask, if extinction by violence is so unlikely, what really happened to the Neanderthals, and how much of them survives in us today? Archaeological evidence does not support the theory that Neanderthals were wiped out by violence from modern humans. Systematic surveys of Neanderthal remains show that less than 5% display injuries consistent with combat or weapon trauma, a rate no higher than what is found in early modern human fossils from the same period. Most Neanderthal injuries are explained by accidents, such as falls or hunting mishaps, rather than interpersonal violence. There are no archaeological sites with layers showing mass killings, no clusters of bodies with weapon wounds, and no evidence of large-scale massacres. A review of more than 200 Middle and Upper Paleolithic sites across Europe and Western Asia finds only isolated cases of perimortem trauma, typically blunt force or single sharp force injuries. For example, the El Sidron site in Spain, which contains the remains of at least 12 Neanderthals, shows evidence of cannibalism but not organized conflict or external attack. Comparative studies of trauma patterns reveal that Neanderthals and early Homo sapiens faced similar risks from their environments, not from each other. The absence of a spike in violent injuries during periods of overlap further undermines the replacement by war hypothesis. These findings are reinforced by a survey conducted in 2021 of 216 paleoanthropologists, in which only 5% identified violent conflict as a primary driver of Neanderthal extinction. The archaeological record, therefore, points away from genocide or competition as the main explanation, opening the way for demographic and genetic factors to take center stage. Neanderthal extinction is best explained through demographic vulnerability, not violence or rapid environmental collapse. The key metric is effective population size, or NE, which reflects the number of individuals in a population who actually contribute genes to the next generation. For Neanderthals, genetic studies consistently estimate NE in the low thousands, typically between 1,000 and 3,000 individuals, across most of their range and for most of their history. Such small effective sizes expose populations to powerful genetic drift, where random chance, rather than natural selection, determines which genetic variants persist. In small groups, drift can quickly erase unique genetic traits, especially when combined with even modest levels of gene flow from larger populations. Mathematical models show that with Neanderthal NE around 2,000 and modern human NE more than double that, the odds of Neanderthal-specific genes surviving drop sharply. If just 1 to 5 modern humans joined a Neanderthal group each generation, their genes would begin to replace Neanderthal variants through drift alone. Over thousands of years, this process can lead to near total genetic absorption, even in the absence of selective advantage or direct competition. The LE effect further compounds this risk. When group sizes fall below a certain threshold, finding mates becomes difficult, reproduction slows, and the risk of local extinction rises. Neanderthal social structure likely involved small, scattered bands, making them especially vulnerable. These demographic realities mean that Neanderthal populations could not maintain their genetic identity indefinitely, even without catastrophic events. Instead, gradual genetic swamping by larger, more connected modern human populations became statistically inevitable. High-coverage sequencing of Neanderthal genomes has revealed that Neanderthals themselves carried substantial amounts of modern human DNA. The Villania and colleagues' dataset from 2024 established that between 2.5% and 3.7% of the DNA in late Neanderthal individuals derives from Homo sapiens, a figure that matches or exceeds the Neanderthal ancestry found in most present-day non-Africans. This genetic signature is not the result of a single event, but of sustained, reciprocal gene flow over thousands of years. D-statistics and segment length analysis pinpoint a major pulse of modern human introgression into Neanderthal populations beginning around 47,000 years ago and lasting approximately 7,000 years. The average length of these modern derived segments in Neanderthal genomes, about 4.2 centimorgans, fits with a mixing window of roughly 100 generations, consistent with the archaeological record of overlapping toolkits and symbolic artifacts during the Upper Paleolithic transition. The distribution of modern human DNA in Neanderthal genomes is non-random. Segments are enriched near genes involved in immunity, skin pigmentation, and metabolism, suggesting that some modern alleles conferred a selective advantage in Eurasian environments. At the same time, certain genomic regions, especially those near brain development genes and on the X chromosome, show deserts of modern human ancestry, indicating that selection also acted to remove incompatible or deleterious variants. The presence of 2.5% to 3.7% modern human ancestry in Neanderthals provides direct evidence for long-term, bidirectional contact between the two populations, with gene flow occurring repeatedly rather than as a single isolated episode. This reciprocal mixing challenges older models of strict separation or one-way replacement and demonstrates that Neanderthals were not simply passive recipients of genetic material, but active participants in a complex web of interbreeding and assimilation. The Villanilla research team's findings set the stage for understanding how demographic imbalance and continuous gene flow, not violence or catastrophic events, drove the gradual absorption of Neanderthals into the expanding modern human population. The Thorin genome provides the clearest single-case evidence of extreme isolation in late Neanderthal populations. Sequenced from a specimen dated to approximately 50,000 years ago, this individual's DNA reveals a total of 180 megabases of runs of homozygosity, which means that nearly 7% of the genome consists of long, uninterrupted stretches inherited identically from both parents. The average length of these segments is about 2.5 centimorgans, far higher than what is seen in any living human population. The inbreeding coefficient, a measure known as F, is calculated at 0.10 for Thorin, more than double the highest values found in contemporary hunter-gatherers, who typically fall between 0.02 and 0.04. These numbers indicate repeated mating among close relatives, consistent with a group size so small that most potential partners were already kin. The Thorin genome team, publishing in Cell Genomics in 2024, compared these findings to other late Neanderthal genomes and found a clear trend of increasing runs of homozygosity and rising inbreeding coefficients as the population declined. Such genetic signatures point to a severe lack of new genetic input and progressive social fragmentation. In practical terms, these levels of inbreeding heighten the risk of harmful mutations becoming expressed and reduce overall fitness. The Thorin case translates abstract demographic fragility into the direct evidence of a single genome, demonstrating that by the time modern humans arrived in force, Neanderthal groups were already so isolated that their genetic line was effectively unsustainable without outside assimilation. A concentrated pulse of Neanderthal DNA entered the ancestors of all non-African modern humans starting around 47,000 years ago. Genome analysts have pinpointed this event using the segment length decay method, which measures how long stretches of Neanderthal DNA persist in human genomes before being broken up by recombination each generation. The length and distribution of these segments act as a genetic clock, allowing researchers to date the main episode of Neanderthal to human gene flow with high precision. The data show that this introgression pulse lasted about 7,000 years, a relatively brief window in evolutionary terms. During this period, repeated interbreeding events transferred Neanderthal genetic material into the expanding modern human population. Today, every person of non-African descent carries between 1 and 2% Neanderthal DNA as a direct legacy of this pulse. The calibration of the recombination clock is based on well-established mutation rates and the observed decay of Neanderthal DNA blocks in living people. The results are consistent across multiple studies and independent data sets. This concentrated gene flow event is not a diffuse, ongoing process, but a sharply defined episode that matches the archaeological record of modern humans spreading into Eurasia. The magnitude of Neanderthal ancestry in present-day non-Africans, always between 1 and 2%, reflects both the scale of the original mixing and the subsequent dilution by a much larger modern human population. The segment length decay method thus provides a precise anchor for the timing and scale of Neanderthal genetic absorption, directly linking ancient DNA evidence to the broader demographic assimilation model. Kolodny and Feldman's neutral drift absorption model demonstrates that Neanderthal genetic disappearance can be accounted for by population size imbalance and low-level, recurring gene flow from modern humans without invoking selection, violence, or environmental catastrophe. The model assumes two populations, Neanderthals, with an effective population size of roughly 2,000, and modern humans with effective population sizes between 5,000 and 10,000. Each generation, 1 to 5 modern human migrants enter Neanderthal groups, an average migration fraction of 0.002 to 0.004. Under these conditions, Simulations show that the probability of modern human genes eventually reaching fixation in Neanderthal populations is between 70 and 85%. The expected time for this absorption process to erase nearly all Neanderthal-specific alleles is 10,000 to 30,000 years, matching the archaeological window for Neanderthal disappearance. The absorption timescale depends on the square of the Neanderthal effective population size. As that effective population size shrinks, the replacement process accelerates dramatically. For example, having the effective population size from 2,000 to 1,000 reduces the absorption time by a factor of 4. This square law relationship means that even modest reductions in Neanderthal group size, through isolation or inbreeding, make genetic swamping by modern humans almost inevitable. The model does not require any selective advantage for modern human alleles. Demographic imbalance alone drives the outcome. Over thousands of generations, the cumulative effect of repeated, low-level migration events and random genetic drift leads to the near total loss of Neanderthal genetic identity. This mechanistic framework provides a statistical explanation for Neanderthal genetic absorption, independent of competition or catastrophe, and aligns with observed patterns of gene flow and allele loss in ancient DNA. The rate at which Neanderthal-specific genetic variants disappeared can be traced through generation-by-generation changes in allele frequencies. Mathematical models, based on neutral drift and recurrent gene flow, show that with an effective Neanderthal population size of about 2,000 and just 1 to 5 modern human migrants per generation, nearly all Neanderthal-specific alleles are diluted to near undetectability within 200 to 300 generations. At an average generation time of 30 years, this corresponds to roughly 6,000 to 9,000 years. The process is not linear. Initial generations see slow, stochastic fluctuations, but as the proportion of modern human DNA rises, the remaining Neanderthal alleles are lost more quickly. This acceleration is driven by what population geneticists call the square law relationship. When the effective population size drops, the rate of allele loss increases dramatically. For halving example, halving the effective population size from 2,000 to 1,000 cuts the absorption time by a factor of 4. Over time, recombination breaks up long stretches of Neanderthal DNA into shorter fragments, further scattering their genetic legacy and making it harder for distinct Neanderthal variants to persist. This fragmentation, known as tract erosion, means that after a few hundred generations, Neanderthal ancestry survives only in small, scattered segments rather than in cohesive genetic blocks. The pace and pattern of this genetic erosion match both the archaeological disappearance of Neanderthal cultural layers and the observed distribution of archaic DNA in modern genomes. These results demonstrate that, given the demographic imbalance and continuous gene flow, the disappearance of Neanderthal-specific alleles was not only likely, but mathematically inevitable, even in the absence of selection or external catastrophe. Neanderthal extinction cannot be explained without considering the biological cost of small, isolated populations. Genome sequencing reveals that Neanderthals carried about 13% more weakly deleterious mutations than contemporary modern humans. This excess genetic load resulted in an estimated 2 to 3% reduction in reproductive fitness per generation, based on population genetic models. The mechanism behind this decline is known as Muller's ratchet, a process where, in small populations, harmful mutations accumulate over time because there are not enough individuals for natural selection to efficiently remove them. As Neanderthal group sizes dwindled, inbreeding became common, further increasing the probability that these mutations became homozygous and expressed their negative effects. The Thorin genome, with an inbreeding coefficient of 0.10 and long runs of homozygosity, exemplifies how repeated mating among close relatives amplified the genetic burden. In practical terms, this meant rising rates of developmental disorders, lower fertility, and increased infant mortality. The combined effect of genetic drift and Muller's ratchet made it increasingly difficult for Neanderthal populations to sustain themselves even before any contact with modern humans. When gene flow from larger, genetically healthier modern human populations began, the influx of new alleles could temporarily mask some of these harmful mutations, but the fundamental imbalance remained. The result was a population that became less viable with each passing generation, accelerating the loss of distinct Neanderthal genetic identity. This fitness decline, driven by mutation accumulation in small, isolated groups, is a central reason why Neanderthals could not persist as a separate lineage once sustained contact with modern humans began. Genomic analysis shows that the Neanderthal legacy in modern humans is not uniform across the genome. Segments inherited from Neanderthals are concentrated near genes involved in immunity, skin biology, and metabolism. These regions, such as the OAS gene cluster and keratin-associated loci, remain common in non-African populations, reflecting adaptive introgression, where Neanderthal variants helped early modern humans survive Eurasian pathogens and environmental conditions. In contrast, large stretches of the genome, especially those linked to brain development and the X chromosome, show pronounced deserts of Neanderthal ancestry. These deserts are the result of negative selection, which rapidly removed incompatible or harmful Neanderthal alleles from the gene pool. The pattern is clear. Natural selection filtered the Neanderthal contribution, retaining beneficial variants while eliminating those that reduced fitness. This selective retention and purging contradicts simple narratives of cognitive superiority or violent replacement, and instead points to a complex process of genetic integration shaped by functional compatibility. Genetic evidence decisively rejects the main alternative extinction theories. DNA analysis finds no genetic signature of sudden population collapse, selective sweeps, or replacement events expected from genocide, epidemic disease, or rapid climate-driven extinction. Models show that if Neanderthals had been wiped out by violence or disease, their DNA would vanish abruptly from the record. Yet Neanderthal ancestry persists in all non-African populations today. The same applies to claims of rapid cognitive superiority. If Neanderthals were outcompeted by smarter rivals, natural selection would have purged their DNA from regions affecting brain development. Instead, negative selection only removed incompatible variants, not the entire Neanderthal contribution. In a 2021 survey of 216 paleoanthropologists, 71% ranked demography as the top extinction factor, with only 5% citing violence and 6% citing disease. By 2025, expert consensus holds that gradual genetic absorption, driven by small effective population sizes and sustained gene flow, is the primary explanation. Hundreds of millions of people today carry Neanderthal DNA, confirming that assimilation, not catastrophic replacement, accounts for their genetic legacy. Today, at least 40% of the Neanderthal genome survives in fragments across modern humans. Clear proof that extinction was not erasure, but absorption. As genome sequencing accelerates, the legacy of small, isolated populations facing demographic collapse is written into our DNA. The real lesson? Human ancestry is not a story of conquest, but of merging lineages. Our evolution is ongoing, shaped as much by loss as by inheritance.